This is chapter 9, section 1. So, we're going to talk about glycolysis, which is a great example of how we regulate metabolism in general in the body. And so, we're going to start with an overview of metabolism, then give a little review about sugars, because that's what glycolysis is: glycolysis. Glyco meaning sugar, lysis meaning break. So, we'll really talk about how the sugars are structured and how we're going to metabolize them. And then the rest is super specific to glycolysis. All right, so we really want to talk about how redox mechanisms account for all of our energy conversion reactions inside the cell. That's a big thing. So most of metabolism is redox reactions, right? And we're converting some kind of chemical energy that we eat into work. And that work for our cells is either catabolism or anabolism. So remember, making and breaking. But here's the thing about catabolism and anabolism. These pathways are active in any given cell at the same time. And so lots of different metabolites serve as both the substrate and the products for different enzymes. So just be aware of that, and that's going to have a lot of implications. And so we'll talk in this chapter about a lot of different catabolic processes. But the anabolic processes, if you want to learn about those, you're going to have to take biochem too, because those are all in the later chapters in the book, and that's what we'll really cover in biochem too. It's really cool. All right, so this is just kind of an example of what happens to our metabolites. So we're going to look at 4 different pathways. We're going to talk about degradation and synthesis of glycogen and degradation and synthesis of glucose. So glycolysis and gluconeogenesis. These are 4 pathways that are active in liver cells all the time. But the relative flux through each of the pathways is going to change in response to glucose concentration, in response to hormone activation of key enzymes, right? Those kind of things. So remember, we're in the liver, and we need to think about what's going to happen to each of our different metabolites depending on the different conditions. And so think about what happens before breakfast, right? You haven't eaten in a while. So tell me about your blood sugar. Your blood sugar is going to be very low. This is like fasting, right? You're going to have low blood sugar. And so what your body's going to say is, well, I'm not influxing very much glucose into the cell at all, right? That's very small. But our glycogen is stored in the liver, and we're going to metabolize it in order to have a higher blood sugar. So we're going to go through a degradation pathway, and we're going to make glucose and send it out for the brain, which is great because the brain needs glucose. And so you're going to, at the same time, promote degradation of glycogen, and you're going to inhibit synthesis, right? So every one of these pathways is like a seesaw. When one goes up, the other goes down. Then at the same time that you're making glucose, do you want to break that glucose down? Do you want to make glucose? Well, I don't really want to break it down, right? It's low. I don't want to do that. I don't want to go through glycolysis. What I'm going to do if I continue to fast is to go through gluconeogenesis. I'm going to go this way because I need to make glucose. So if my glycogen stores run out, I got to make glucose somehow, and I'll go through gluconeogenesis. But the end result is we're going to send out glucose. We're going to efflux glucose into the bloodstream to meet the needs of the cell, right? So think about what's catabolism here and what's anabolism here. Anabolism is our gluconeogenesis. Anabolism is synthesis of glycogen. Degradation is catabolism, right? So is glycolysis. So every one of the catabolic pathways has a like mirror image almost anabolism. So just keep in mind. Okay, so let's look at the next one. The next one's after breakfast. We just ate. So this is like the fed state. We just ate. So we had a huge influx of glucose into our bloodstream, right? So we don't really need to make glucose anymore. We have a lot of glucose. We're going to go through glycolysis. We're going to break down that glucose, make pyruvate, and pyruvate goes off to be used in lots of different things. And the other thing that we can do with excess glucose is that we can go and make glycogen for those times when we don't have a lot. And so those are the two sides of the coin that are going to be upregulated. But at the same time, while we're making glycogen, we want to inhibit degradation of glycogen and we want to inhibit gluconeogenesis because we have a lot of glucose. So we're going to keep all this in mind when we're trying to balance our different pathways. So how do we regulate all of this? Well, the level of enzyme activity is one. So how can we regulate levels of enzyme activity? Think about gene transcription. Think about protein synthesis. Protein, it's synthesis, it's turnover, right? That's degradation. Think about catalytic activity. Think about cellular location, right? Cellular, UL. Ooh. Cellular location. You're going to also think about binding of regulatory molecules. Running out of room. Okay. All of these different things, covalent modifications, right? All of those things are ways for us to regulate enzyme activities. There are lots and lots of ways that the cell regulates enzyme activity. Then talking about levels of substrate, that is going to have a major effect on which pathways we're going to go through. And we see that just in glucose, right? Do we obtain it from the diet or is it released from our energy stores in the body? Whichever place that we get it from, it's going to have an effect on how we metabolize it. So I want to show you this. So even though what we are going to do, we're going to look at individual pathways, you have to understand that one pathway is highly interdependent on lots and lots and lots of different pathways, right? They're controlled by enzyme activity levels. They're controlled by our substrate bioavailability. They're so interconnected. So I don't show you this to scare you. I'm just showing you this so that you have this deeper understanding of how things are connected. And so this is a little database that shows the linkages of substrates to products using, it's like an interactive grid, and each dot's a primary metabolite, and the lines correspond to series of enzymatic reactions. But it's too much detail to study all at once, right? So what we want to do is we want to study, and I call this studying the trees in the forest, right? We're going to examine one pathway at a time because it's easy to learn that way. But really, the key to understanding metabolic integration in terms of nutrition and exercise and disease is to learn how our cells control metabolic flux between pathways. And so you have to keep in mind that the collection of metabolic pathways in an entire organism is super complex. So there is a big forest, but we're only going to study one tree at a time. This is the map that we're going to study. This is a much more simplified version, right? And so it's important to understand this simplified version, okay? So we have 4 major classes of our macromolecules. That's proteins, nucleic acids, carbs, and lipids. Now, as those are metabolized, we have some primary metabolites. That's the blue. The blue are all primary metabolites. So primary metabolites are things that our macromolecules can turn into. Okay. Then we have these ones down here. It's kind of reddish. These are the 7 small biomolecules, right, that are going to be really, really important. And so we're going to kind of focus on these in the simplified version, okay? Simplified version. So what we're going to do is we're going to divide this metabolic map into 2 distinct pathway groups. And then the pathway groups are going to be subdivided. But, okay, what does each one mean? So the red ones, these red ones are energy conversion while we are oxidizing simple sugars. Energy conversion. We are oxidizing simple sugars. And remember, I told you it's redox reactions. That is what is going to drive metabolism. So this is redox reactions. And what we want to eventually accomplish is generation of ATP. And it's really this. Really, this is what we're going to study. This is what we're going to do from now until the end of the year. Just this little part of the pathway. Then look at the blue. You need to understand the blue. And the blue are pathways that control synthesis and degradation of our different macromolecules, right? So we're not going to have time to work on all of those this semester, but we will work on them next semester in Biochem 2. So if you're going pre-med, if you're going pre-vet, if you're doing those kinds of things, you really need that. Okay, so before we can really look at glycolytic pathways in detail, we need to revisit the concept of bioenergetics that we covered in chapter 2. So we're going to look more closely at how the concentration of substrates and products affect our free energy, our Gibbs free energy of coupled reactions. So one thing you really need to remember is what are our standard conditions, our biochemical standard conditions, because that's what we're going to be looking at for every reaction. We don't really care about standard free energy. We care about biochemical standard free energy. So remember, we're at 37 degrees, right? We're at about pH 7, all of those things. So go back to your notes and go look that up. But the other thing to remember is that when we calculate delta G naught prime, this is at equilibrium. And we're not necessarily at equilibrium for any of our reactions, right? And so if you want a reaction to go, it has to have a negative delta G value. That's what makes it spontaneous. But if it doesn't have a negative delta G value, that doesn't mean that the reaction won't go. It just means that that reaction is unfavorable and we're going to have to change the conditions in order to make it go. So the way that we do that a lot of times is to couple reactions where they use that shared intermediate. And we see that a lot when we couple it with ATP hydrolysis, right? So what's happening is that we're going to use our energy that's stored in ATP in order to drive the reaction, right? So sometimes we actually want to go in the other direction, right? So sometimes we want to make ATP. And so that's what we're really going to be talking about in glycolysis, is we're going to be making ATP. So we're going to use reactions that have extremely negative values in order to drive a positive delta G of making ATP. And so eventually it will happen and we will make ATP because our net reaction is a negative delta G value. And so because we have this negative delta G value, both of these reactions are going to proceed in the forward direction. And you have to remember that all of these reactions are made possible because you're using really, really highly specific enzymes that are going to harness the free energy that's released in small increments, right? So, like in the very first step of glycolysis, it's going to be catalyzed by an enzyme called hexokinase. Hexokinase is going to use ATP hydrolysis to drive the phosphorylation of your glucose molecule. But we can also couple reactions to capture free energy in the form of ATP, like this example right here that we have, where we are actually capturing that free energy in the form of ATP. And this is the very last step in glycolysis. So it can go either way. So you need to know, how can you calculate metabolic flux? It's not really delta G naught prime conditions. It's not. We want to calculate delta G overall changes because we want to know at actual concentrations of the substrates, what is my delta G value? So you have to remember that we're not calculating equilibrium concentrations. We're calculating Q, which is actual conditions. So if we take a look at one example, here's our one example. We have glucose 6-phosphate. So a lot of times we abbreviate it G6P, glucose 6-phosphate going to fructose 6-phosphate. This has a positive delta G of 1.7. So it's unfavorable at standard biological conditions. Okay. Now, if we look at this and say, well, what are the actual concentrations of each of the substrates? If I plug that into Q and I solve for the actual delta G value and do all the math, I get an actual delta G value of negative 2.9. So while this reaction is unfavorable at standard conditions, in the cell, we're not at standard conditions. We are at biological conditions. And so at biological conditions, we have a negative delta G value. So our reaction will proceed in the forward direction. So you're going to see this over and over and over again. If you look in a table and you see in the table that our delta G naught prime is positive, as long as it's just a little positive, then we're not going to worry about it too much, because we can have a lowly positive delta G and the reaction will still go forward because it is coupled to something else, or we change the concentrations and that changes how the reaction proceeds. Okay, so our reason why this works is because we have these shared intermediates. And so as we change the concentration of one of our substrates, so here's fructose 6-phosphate, this is a very low concentration. And so because we have a low concentration, that's what actually allowed us to have a negative delta G. So think about this in terms of biochemical pathways it's a chain of events, right? You're going to have glucose 6-phosphate, and that's going to go to fructose 6-phosphate. But then we have another step after that. We're going to go to fructose 1,6-bisphosphate. So if I deplete some of my fructose 6-phosphate because it's being used in the next reaction, I end up with a low concentration of it, which would cause the first reaction to make more product. So it's just making sure that we can drive those reactions in the direction we want to. So remember that our enzymes are not just floating in solution, right? It's really typically large multi-protein complexes. And because you have really close interaction between these enzymes, you're limiting product diffusion. So our product's not being made and floating off. It's channeled as an intermediate into the very next enzyme. And so you'll you're going to make sure to drive that. Okay, structures of simple sugars. Remember, we talked about glycolysis, and we said glyco meaning sweet, lysis meaning to split. So glycolysis is the splitting of one glucose into two molecules of pyruvate. And so you have to know what glucose is in order to understand then what pyruvate is and all that kind of stuff. So if you have one sugar, it's a monosaccharide. That monosaccharide can contain either an aldehyde, so then it would be an aldose, the OSE tells you it's a sugar, or it can contain a ketone. So remember, your aldehyde looks like it's all alone. That's what I call it. I call it all alone. It's only one R group. But your ketone, on the other hand, has two R groups. So a ketone, R1, R2, needs two things, a lock and a key. An aldehyde is all alone, so it only has one R group. The other thing attached to the carbonyl is just a hydrogen. Okay. So if we look at glucose, glucose is a polyhydroxyl. So it's got hydroxyls, OH, all over the place, aldehyde. Fructose is a polyhydroxy ketone. So they both have the formula C6H12O6, but they're not the same because one of them is an aldehyde, one of them is a ketone, and that's very, very important. But sugars in general taste sweet, and we find them naturally in the foods that we eat, fruits, vegetables, all that kind of stuff. So why do they taste sweet? It's a result of a ligand binding to a G protein coupled receptor, remember that, and they bind to different sugars with different affinities. So not all sugars taste as sweet as each other. So for example, fructose has more than double the sweetness as glucose. So that's why you'll see manufacturers use high fructose corn syrup, because they can put in less and it'll still taste just as sweet as half as much glucose being put in. So if we look at some simple structures, glucose and fructose, glucose we said was an aldehyde. So here's your aldehyde. And remember, everywhere where we have an intersection of lines, that is telling us that we have a carbon right there. So if you have anything that is a perfect plus sign, that's telling you that it's a chiral carbon, which means it has four different things attached to it. So here are carbons 2, 3, 4, and 5 are all chiral. Okay. Carbon 1 is not chiral because it only has 3 things attached to it. And carbon 6 is not chiral because it has 2 hydrogens attached to it. Okay. So we can look at sugars in one of two forms, as straight chains or as rings, and they kind of fluctuate in between, right? And it happens spontaneously. So how do you go from a straight chain to being a cyclic molecule? So when we look at a straight chain, what we see is that our hydroxyl on our very last chiral carbon is going to come up, the oxygen is going to come up, and it's going to make a bond to the carbon that is involved in the carbonyl. And so the oxygen, this oxygen right here, becomes this oxygen right here. So carbon 1, which was the carbonyl, is now inside the ring, and we call this the anomeric carbon. So the anomeric carbon is always going to be bonded to 2 oxygens, right? And so here we see 1 oxygen is outside of the ring, 1 oxygen is inside of the ring. So you can kind of see that. Okay. Then when you're also drawing this, in order to know what conformation this ring is in, right? Remember, rings have things above the ring and below the ring. So you have to, and you can't break the ring. So once they're above or below, they're stuck in that position. So if we look here, we know this hydroxyl is going down, it's below the ring, and our largest numbered carbon outside of the ring, which is carbon 6, is going up. So if we have it going up and down, you know how like double bonds are cis or trans? Well, rings can be cis or trans, right? So this is trans because one's going up, one's going down, but in sugars, we don't call it that. We call this the alpha anomer, right? So the same thing, we can look at that in fructose. So here's our anomeric carbon because it's bonded to 2 oxygens. My hydroxyl is going down, my C6 is going up. So this is also the alpha anomer. Okay. But just be careful when you're numbering your carbons on anything that's a ketone. Because it's a ketone, my carbonyl carbon is not carbon number 1. So when this oxygen comes up and binds to this carbon in order to make the ring, you're going to have one carbon that's outside the ring that's attached to your anomeric carbon. And so your anomeric carbon is going to get the lowest number possible. But if you have a carbon that's outside of the ring, that gets carbon 1, then your anomeric gets carbon 2. If you have an anomeric that does not have a carbon outside the ring, then it gets number 1. So just be careful about that, right? So when we look at things that have chiral centers, right? Their chirality is important, and our enzymes are so super specific. They can tell the difference between just having those groups switched. So we call them D or L. Remember, we talked about this with amino acids, and that amino acids tend to be L, whereas our sugars tend to be D. So how do you know if it's D or L? You are going to look at the hydroxyl that's attached to the carbon at the penultimate carbon. What's the penultimate carbon? It's just the last chiral carbon. This is the one that's going to go up and bond to our carbon in the carbonyl to make the anomeric carbon. It's the same one. So if it's on the left, we call it L. If it's on the right, we call it D. So this one is on the right, so we call it D-glucose, right? So just remember that every single compound that has a chiral center, right, that has a plane of symmetry, it can exist as 2 optical isomers, and we call them enantiomers. So if I wanted to draw the enantiomer of D-glucose, I would draw the perfect mirror image of D-glucose. So, right, it doesn't matter at the top because these aren't chiral, so it really doesn't matter which way that you draw them, right? And then you have CH2OH, and we have chiral center, chiral center, chiral center, chiral center. So all we do is swap them, right? So you swap everything. What was on the left is on the right. So on this side, I'm going to go hydrogen. On this side, I'm going to go hydroxyl, hydrogen, hydrogen. Then on this side, I'm going to go hydroxyl, hydroxyl, hydrogen, and then hydroxyl, right? So this is just L-glucose. Not a big deal. All right. So then you can have things that are epimers. Epimers differ in one position on one chiral carbon. So if you look at D-glucose, and this is what a lot of our different sugars are. If I just make a C, so here's my first carbon, 1, 2, 3, 4, 5, and then this is 6. So this, to go from D-glucose to D-mannose, it's a carbon 3 epimer. D-mannose is a carbon 3 epimer of D-glucose. All we did was take the hydroxyl and the hydrogen and swap them, hydroxyl and hydrogen. That's it. So they're aldohexose epimers. D-ribose and D-xylose. All we're doing here is a carbon 1, 2, 3. That's a C3 epimer, right? So they're aldopentose C3 epimers. Not a big deal. All right, Hayworth projections. These are the rings. So this is the reaction I was telling you about where they go through that intermolecular reaction to form a ring. And depending on how many carbons are in the ring, we either call it a pyranose or a furanose ring. So the ring is more stable than the straight chain. And you're going to have to remember just those naming rules that I told you about, right? So just like I told you before, this oxygen right here is going to come up and it's going to make a bond with the carbon in the carbonyl. Okay, so what was the carbonyl carbon, that's the one that's going to make this bond. So the green line that I drew over here is the same as the green line I drew here. You just have to remember that the atoms don't have to stay straight. They can kind of wrap around. So the oxygen is going to make a bond with the carbon. Carbon does not want to have 5 bonds, so you're going to lose one of the double bonds. Then this hydrogen is going to go over and bond to this oxygen. So we'll get a hydroxyl, right? Where we did have our carbonyl, we'll get a hydroxyl. And so if we look, here's our hydroxyl that we just made, that hydroxyl right here, that hydroxyl right there. And then this oxygen is this oxygen. So what was the carbonyl becomes the anomeric. So this was my carbon that was in the carbonyl, and now this is that carbon. So what was the carbonyl becomes the anomeric. And what was the hydroxyl that determined DRL, that is the one that is now in the ring. Same one here. Okay. So one of two things can happen when that cyclic reaction happens. We can either have the hydroxyl going up or the hydroxyl going down. And actually both reactions form. And how do we know which one is which? Okay, let me kind of erase some of this stuff. Here we go. So remember we said we take our anomeric carbon, we look at the hydroxyl, and we look at C6 going outside the ring. They're opposite, so that is trans, and we call that alpha. And then the other way that that could happen is the hydroxyl could end up going up, carbon's going up, and we call that beta. Beta is actually a little more prevalent, but we call that our pyranose forms, right? And so the only difference between our alpha and beta anomers are the position of the hydroxyl on the anomeric carbon. It's not a big deal. Okay, so when we look at sugars, sugars can be reducing sugars or non-reducing sugars. And so when we talk about these sugars, you have to look at their structure to know if they're reducing sugars or not. And so things that are reducing sugars will react with oxidizing agents. So if we have something like an aldose is able to react with, for example, copper. So remember when we talk about oxidation reduction, oil, rig, right? Oxidation is loss of electrons, gain oxygen, loss hydrogen. You're going to gain electrons, gain hydrogen, loss oxygen. Okay. So what happens when we go from copper 2+ to Cu+? So we're reducing our copper because our copper is gaining electrons. Its charge is going down, so it's gaining electrons. And then our sugar, if we look at our sugar, what is happening when we go from D-glucose to D-gluconate? Well, look at your carbon that's in the carbonyl. What happened? I converted that hydrogen into an oxygen, right? So I am gaining oxygen and I'm losing hydrogen. So that is an oxidation of our sugar, right? So they call this Benedict's test. And you can see this because our Cu2+, if you had it in a beaker, in a test tube, whatever, it's blue. It's a blue solution. but then when it goes to Cu plus, it turns into a red solution. So you can very, very easily see the color change. But there is a new test nowadays. Nowadays, they don't use that test. Nowadays, they do a test. I don't think I gave you the thing on it, but you have glucose, right? And you're going to convert it to gluconate. And gluconate, if I can move this, there we go. All right, so you have to have that oxidation, but in the new test, it's just oxygen that converts it into H2O2, right? So this is called glucose oxidase. So you have this enzyme, glucose oxidase, that converts the glucose to gluconate. And then you have this dye. So in the presence of hydrogen peroxide, H2O2, right, your dye goes from the reduced form to being an oxidized, you don't have to know the thing, but to an oxidized form. And this is peroxidase that does this. And so this is the new test. It's not Benedict's test anymore, but this is the new, like the blood sugar test, you know, the little ones where you prick your finger and you're able to do it really quickly in that little bitty thing, right? That's how we do it. So like when you eat, right, your blood sugar goes up, your hormone insulin's produced, your cells pick up the glucose, and so your blood sugar drops. But in people with diabetes that have improper insulin signaling, whichever way it is, so it results in high blood sugar levels. Like when we're talking like high, like 130 milligrams per deciliter, that's pretty high. So how do you know that it's high? How do they measure their blood sugar? So the aldose sugars in an open chain, right? Those are the ones that can be oxidized. And so we go from having this aldehyde to having a carboxylic acid. And so that's the key for being able to determine what's your blood sugar level, right? Okay, so a lot of times when we eat things, we don't just eat monosaccharides, we eat disaccharides. So disaccharides are two monosaccharides that are linked together with an O glycosidic bond. And we'll talk about the structure in just a second. The reaction that makes a disaccharide is a condensation reaction. So remember, condensation, you are making water. So lactose is a common disaccharide. You need to know this. It's galactose in a beta 1 to 4 glycosidic bond with glucose. Then sucrose is glucose in an alpha 1,4 bond with fructose. And that is actually table sugar, right? That's table sugar. Trehalose, deathlands in insects, that's a glucose, CO, glucose, and that's a alpha 1 to 1. It's alpha 1, I think, glucose. Yeah. So when we talk about that, how are we giving these designations, right? Okay, if you look at an individual, here's lactose. Lactose, you need to know lactose. Okay, it's an O glycosidic bond because I have an oxygen and it's bound to one sugar, two sugars. That's my glycosidic bond, right? Okay, so now, how do I know that this is a beta 1,4 glycosidic bond? First thing that you do is I want you to find your anomeric carbons. Anomeric carbons are bonded to 2 oxygens, okay? So I have 1 anomeric carbon on every sugar. There's my 2 anomeric carbons. Any anomeric carbons that participate in the glycosidic bond, you need to tell if they're in the alpha or beta configuration. So I only have to do this for one of them. So I'm going to call this sugar A, sugar B, right? So for sugar A, we need to determine the orientation of the bond. So this one's going up, this one's going up. So up and up is beta. And we don't need to do that for the other one. The other thing that we need to do is number our carbons. So the anomeric carbon gets carbon number 1 unless it has a carbon bonded to it outside of the ring. And we don't see that here in A. So that's 1, 2, 3, 4, 5, 6. Then on sugar B, Anomeric gets 1, 2, 3, 4, 5, 6. So now, which carbons participate? Carbon 1 and carbon 4. So we call this a beta 1 to 4 glycosidic bond between galactose and glucose. Okay, so try the same thing. You try on this one. So first thing is to find our anomeric carbons. So anomeric, and then we're looking for anomeric. I hope that didn't trip you up. Then we have A and B. So I have 2 anomeric carbons that participate. So the 2 anomeric carbons, I have to give alpha and beta for each of them. So here I'm going down, here I'm going up. Down and up gives me alpha. I have to do the same thing for my other anomeric carbon. So I have my glycosidic bond going down, then my highest numbered carbon outside of the ring. And you might have to number them first, that's okay. So my anomeric carbon gets carbon number 1 unless I have another carbon bound to it. I have another carbon bound to it. So it's 1, 2, 3, 4, 5, right? So 6 is there. So now I have my glycosidic bond going down. I have my C6 going down. So that's what gives me beta. So now I just need to know which carbons are participating. So then I'll go, this is carbon number 1 because there's no other carbon attached to it. 2, 3, 4, 5, 6. So which carbons are participating? That is 1 and 2. So alpha 1, beta 2. Okay. You try this one. Okay. So I'm going to find my anomeric carbons. That's 1. Here's the other one. I'm going to say, let's number them. 1, 2, oh, this is A, this is B. So 1, 2, 3, 4, 5, 6. I'm going to number this one. I'll go 1, and I had to go up instead of going down because I'm not going to number my oxygen. I'm going to number my carbons. 2, 3, 4, and then that's 5, C5 right there. Okay, so now I need to know, are we going up or are we going down, right? So glycosidic's going down, carbon 6 is going up, so this one is alpha. Here, glycosidic's going down, and then my highest number carbon outside is the 5, that's going up, so I got a down and up, that's an alpha. Then which carbons are participating? Oh, I numbered that wrong. Y'all, number your anomeric carbon. Hello. Hello. 1, 2, 3, 4, 5, 6. Okay. You should kind of notice that. All right. So here are my carbons that are participating. So what do I have? I have the carbon 1 and carbon 1. The first one is an alpha. The second one's an alpha. So alpha 1, alpha 1. Glucose to glucose. So that's how you do that. So anytime we consume a disaccharide, we have to be able to metabolize it into a monosaccharide. So going from a disaccharide to a monosaccharide is hydrolysis. So we're going to use water in order to hydrolyze it. And basically what happens is that you just take wherever the glycosidic bond was. Watch, I'm going to cover it. So if it was both going down, see how it was going down? All we do to make our products is to add a hydroxyl, right? Because one of the oxygens was in the glycosidic bond, one of the oxygens was from water, and then they each get hydrogens, and so that's my product. It's the same thing. It's like the easiest reaction ever. Okay, so we take maltose, we hydrolyze it, we get glucose. Sucrose, we hydrolyze it, we get glucose and fructose. Lactose, we hydrolyze and we get galactose and glucose. Here's the thing with lactose, right? Anybody know about people who are lactose intolerant? So the human gene for lactose is expressed at super high levels when you're a baby, when you're an infant, right? Because it aids in digestion of breast milk, but its expression declines as we get older. So if you don't if you don't produce the enzyme lactase, you don't metabolize lactose. And so that lactose is then metabolized by bacteria in your gut. And so they will ferment the lactose, and they'll produce H2 and methane, which are gases. And so you will have lots of gas and you will have diarrhea, right? So, oh, yeah. Oh, so here, here it is. So we talked about all that, right? So what do you do? Let's see, did I? No. So what do you do? How do you withstand this terribleness? So you can take like the Lactaid. So that's just a pill that has the enzyme in it, right? And so that will help you to metabolize it because you're ingesting the enzyme as you're eating it. You can buy the milk that has the sugar already hydrolyzed. So if you buy, I don't remember the name brand of the milk, but it'll take the lactose, it'll take the milk and it'll add lactase to it. Then it metabolizes it. So then the milk has glucose and galactose and not lactose in it, right? And I don't think I said, but so why? So you know why you get gas, but why do you get diarrhea? So if you have a high amount of unhydrolyzed lactose, um, lots and lots of water is going to be pulled from your intestinal cells to like hydrate it. And so that's what gives you the diarrhea. But you can take your Lactaid and you'll be fine. All right, that's it. Have a great weekend.